My name is Irina Kissileva. I'm one of the co-authors of the paper and the presentation, so I will do it. First of all, I would like to agree to all the participants of the conference and say many thanks to the organizers for this opportunity to make a presentation at the conference. So. I will speak about the licorice. Licorice, you know, is one of the wonderful plants that are widely used in medicine, and it is traditionally used in many countries. The roots and rhizomes of licorice are used for as the expectorants and emollient. They also used in different kinds of mixtures as a component that improves the taste and penetration of other remedies. Also, the biomass of licorice rhizomes is used as a flavoring additive to the confectionery and food industries. Active harvesting of rhizomes leads to the depletion of natural reserves of this plant, and it's a lack of raw material for industries, for medicine, and for example, in many regions of Russia and neighboring countries, licorice is listed in the red books and need protection to prevent the total extinction of the populations. A modern and effective method for preserving the natural biodiversity of medicinal plants is their introduction into the cell cultures or tissue cultures. However, the difficulty of using this technology lies in the fact that the qualitative and quantitative composition of kali and intact plants often are different. So it is known that the licorice rhizomes contains many active ingredients. For example, saponins, flavonoids, polysaccharides, and so on. One of the most important compounds are steroid saponin as glyceric acid and flavonoid picorazide. They have a very strong biological activity, and they reveal the most biological effects of licorice. So, as the licorice is a very popular plant for medicinal purposes, as we have said, the biomass of this plant is actively used by people, and there is a strict need to take new technologies for obtaining the biologically active substances from the licorice. So the purpose of our work was to obtain callus cultures from leaves of licorice and to study their antioxidant and biological properties, and also to assay phenols and flavonoids in this kali. So as rhizomes are actively studied, so we decided to point out on the leaves, because it is known that they also have a lot of biologically active substances and could be considered as the raw material for the obtaining these biologically active substances, mainly with anti-radical and antioxidant activity. So the licorice plants were taken from the botanical garden of the Ural branch of Russian Academy of Sciences. There at the botanical garden, the plants were introduced from natural populations from the southern Urals. So the leaves of these plants were used as an implant material for the obtaining kali. The plant material was sterilized according to the standard procedure, standard technique, using 70% ethanol and sodium hypochlorite. And for the induction of primary kali, we used the Murasigi Skog medium with sucrose 3% and also phytogormans as BAP the cytokinin and auxin, naphthalene, about the plant material. So as I have mentioned, the plant material for getting calyces were the leaves of licorice, and we used the MS medium with 3% of sucrose and combination of phytogormons with benzylaminopurin and naphthylacetic acid. So different combination of these auxins and cytokinins were used. Okay, so totally it was about 9 combinations of phytogormons, and they were tested in order to understand what ratio is the most optimal for growing kali. So on this picture you can see the kali obtained from the licorice leaves on the medium with different concentrations of benzylaminopurin and naphthylacetic acid. The upper line in each case show us the kali after 1 week of cultivation, and the low line is demonstrating the kali on the 4th week of cultivation. So the analysis of this kali showed us that the best concentrations of phytogormons was 0.221 for cytokinins and 10 to 20 auxins in the media. Here you can see the percentage of the surviving kali in different conditions. So on this slide you can see the total amount of phenols and flavonoids in the extracts obtained from the studied calyces and the intact plant. So in plant leaves, in plant roots and rhizomes, and in calyces grown on different media, we have tested the amount of phenols and flavonoids. Why these substances? It is well known that they have a very good anti-radical and antioxidant activity, and that is why we decided to understand if kali is good for this sense, comparing with leaves or roots. As you can see, leaves were very rich of phenols, much more than roots and rhizomes, and that were tested in calyces. And the flavonoids, about flavonoids, we can see that the amount of flavonoids in roots and leaves were approximately the same, and also in calyces grown on the media with 10 milligrams per liter of auxins. So another type of calyces were low in both in phenols and flavonoids. So we also tested the antioxidant and anti-radical activity of the extracts from the leaf calyces, and it was noticed that in ABTS test, there is no significant difference. There was no significant difference between the extracts of roots and rhizomes, leaves, and 2 variants of calyces. So this test showed that both in intact leaves, rhizomes, and both types of calyces, the extracts could inhibit the ABTS radical formation quite good. So in other tests, we estimated the reducing potential of the extracts from leaves, rhizomes, and calyces. And it was shown that the leaves were the most efficient. The leaf extracts were the most efficient among all the studied extracts. And there was no difference between the reducing potential of calyces and rhizomes that are usually used as raw material for remedies. So, and also we studied the ability to inhibit the nitric oxide formation by these extracts, and it was shown that calyces have a little less activity than leaves and rhizomes, but not very low. And comparing this reducing or antioxidant activity of the extracts with the standard antioxidants as ascorbate, gallate, and rutin, we can understand that calyces are quite efficient for quenching radicals and showing this anti-radical and antioxidant activity. So also reducing potential and inhibition of ABTS radicals and NO radicals could be quenched by these extracts quite good. In our study, we have shown that the optimal conditions for the obtaining calyces from licorice leaves is rather simple and very widespread. It is MS medium with sucrose and a combination of phytogormons, about 1 milligram per liter of cytokinins and 10 milligrams per liter of opsins as naphthylacetic acid, for example. We have shown that the calyces have a good combination of phenols and flavonoids, and compared to intact leaves, they are not. They are also have a lot of these antioxidants extracted from them, and we also can say that the content of phenols and the ratio of the content of phenols and flavonoids in calyces originating from leaves are comparable with the intact roots and rhizomes. So the antioxidant properties of these calyces are also comparable to the original parts, or to the parts of the original plants. Thus, the calyce culture of licorice obtained from leaves can be a promising new raw material for the synthesis of secondary metabolites, in particular flavonoids, with a high antioxidant potential. So this work was done under the supervision of Dr. Alex Yermoshin, and several people took part in these studies, both students and staff of the Ural Federal University. So I would like to thank the colleagues who were involved in this research, and also to thank all of the participants of the conference for your attention. Thank you very much.
If any of the participants has any question, they can unmute themselves and ask the question.
Yeah, I have one question. Myself, Dr. Vineet Maurya.
Okay.
Yeah, the question is when we talk about secondary metabolites, they are produced at the later stage of the growth, and when there is a harsh condition or the adverse conditions, they will be produced much more. So the concentration produced in the by lab tissue culture and the naturally growing in the harsh condition, will it be same or there will be difference? Is there any study like that?
We have studied only the phenols and flavonoids in the extracts of kali, and both in the extracts of leaves and rhizomes of the licorice. We paid attention to these compounds because they have a very good anti-radical and antioxidant activity, and it is well known that any other biologically active compounds from licorice, they have another chemical nature, and we didn't study them in the calyces.
Dr. Kissileva, let me rephrase the question from Dr. Maurya. There are certain environmental triggers which actually incites the plant to produce more of those phytopharmaceuticals. But do you have studied about any of those triggers which can enhance the production of specific pharmaceuticals in the plant cell cultures?
Yes, thank you. So no, we didn't study the triggers. We just studied, the efficiency of adding some phytogormons to the media to obtain best calices, to let them grow fast and have grow fast and have a big mass. So we didn't study any other triggers.